Well, welcome everybody to our 2025 GIST Educational Conference. Here for two purposes. One is GIST Cancer Research Fund and their founders, Robert and Tanya Stetman, are here. They have been longtime donors, like 20 years, I think, to the 24 years to the night. And the second purpose is to provide education for patients and their caretakers about GIST in general. And some of you almost have a PhD in GIST because you've had it so long and been through lots of treatments, and others of you may be more newbies and just this is the beginning of your journey. So gastrointestinal stromal tumors are these somewhat uncommon. I don't like to say rare because if you say rare, nobody will care about it, especially people that you're trying to ask them for grant money. So we say uncommon. So about 6,000 cases that are clinically significant. Most of these arise in the stomach, 60%, but they can arise in the esophagus. That's pretty uncommon, but also in the small intestine or the rectum. And then we can diagnose these endoscopically. So this is an endoscope looking into the stomach, and you see a tumor, which is kind of that fleshy thing coming out here, and it's actually partially ulcerated. And if you remove it, and here's a piece of the stomach, and now the tumor has been cut open, so you can see next to the ruler how big this is in terms of centimeters. And these cells arise in the muscular wall. And so, we think of the stomach as the digestion, right? So that's like the food's in there and we're absorbing the food and grinding it up and all secreting acid, all that kind of stuff. But in the wall are muscles that have to sort of squeeze and contract and mash everything up and then push it the right way. And so those need to be organized. So in here, in the wall of the stomach, are these little things that kind of look like spiders or starfish. And those are the interstitial cells of Cajal, which are pacemaker cells. But those are the cells that when they get corrupted by mutation are what form the GIST. So even though there's a lot going on in the stomach, the cells that give rise to GIST are very specialized and in the wall. And we can actually see normal variants of that by staining them. And so all the blue stuff is the smooth muscle, and the brown staining stuff is actually these interstitial cells of Cajal. Now, we know that GIST are caused by DNA mutations. So I wanted to say a little bit more about what is a DNA mutation and how does that relate. So in terms of a basic understanding of molecular biology or studying DNA mutations, DNA is like the master blueprint. It's the code, and it gets eventually, through processes, turned into protein. And protein is what does the work. And so the protein defines the function of what's going to happen. So you could think about that in terms of language, that DNA is simply a code or series of letters, but we can use those letters to make a sentence, and then the sentence will have a specific meaning if we construct it correctly. And so unlike an alphabet, the English alphabet where we have 26 letters, DNA is a simpler code in a way in that it only has 4 letters, C, A, G, and T, and organized in little groups of 3, but that can have a meaning. So we could say, for example, in a sentence form, the cat is fit, and then when we read that sentence, we know what that means, that the cat is fit.
I'm Christine Robbins. I've been in the lab for now just about a year, and I have been working on SDH deficient GIST specifically. Mutations, as Mike was talking about, in various genes can increase the chance of getting cancers. So here we just have an example of a DNA, which codes a gene, which will make a protein, which will have a function. If something goes wrong in our cells, they are like us, they make mistakes. We can end up with a mutated gene, which can give us a mutated protein, which can alter or hinder, get rid of the function. GIST is caused by a number of different mutations in various genes and proteins. The one I'm going to be talking about today is SDH deficient GIST, which can be a little bit different than the other ones. Mike had mentioned the KIT mutations are like always turned on, so like a car that will not stop. A lot of times, in general, SDH deficient GIST is caused by mutations where the car basically stalls out and stops working, which can make it a little bit trickier to treat with drugs, because most drugs can turn things off, but they have a very hard time getting things to turn on. So figuring out what mutations can cause SDH deficient GIST can be really important for treatment and early detection. But what is this SDH that I am talking about? So SDH stands for succinate dehydrogenase, and this really plays a critical role in letting our cells have metabolism and energy and really kind of gets our cells going to divide and have all their normal functions, all the way up from being people to our tissues, our cells, and our little mitochondria, which if we remember from middle school biology is the powerhouse of the cell that makes all of our energy and keeps us functional. So this is a somewhat complicated diagram of succinate dehydrogenase. It's a complex that is made up of 4 different subunits. In humans, they're SDHA, B, C, and D, and they all work together to provide energy and to help with our metabolism. They kind of have a bit of a dual role. And what happens is, if one of these proteins doesn't work properly, let's say SDHA, which is what I'm going to be talking about, let's say there's a mutation and it no longer works. We're no longer able to get this process, and we end up with a buildup of succinate, which is what we believe causes problems in metabolism in cells that can result in cancer. But not all of the mutations that we have cause problems. As Mike had mentioned, cat with a C versus cat with a K, not really going to be an issue. Those are mutations that we kind of call benign or likely benign, which aren't really likely to be harmful. However, some mutations that we know are pathogenic, can cause diseases, are going to be kind of over on that other side. But there's this big category in the middle of VUS, or variant of uncertain significance, sometimes variant of unknown significance. And we're not really sure what these guys do. They might cause disease, they might not. We kind of haven't figured it out yet. So one way to think about this is if we are walking through, let's say, our lovely forest park here, and we stumble upon a brand new mushroom, and we've never seen this mushroom before. We don't know if we can eat it or not, or it's going to be safe. Can we eat it? Can we put it in our food? Who knows? We want to find out if this mushroom is benign, pathogenic, but right now it's a VUS. So we need to figure out, can we put it on our mushroom pizza? Can we put it in our mushroom tacos? I was hungry when I made these slides. Or is it something that's going to make us a little bit sick? So in general, in the lab, what we try to do is scientists, we need to find out if these mushrooms or mutations are going to be pathogenic or benign. So this is really critical, again, for better diagnostic tools, early detection, and kind of figuring out what's going on as early as we can, because a lot of the drugs that we have don't necessarily work as well on SDH deficient GIST. So what I do is I actually use the budding yeast, Saccharomyces cerevisiae, in order to kind of answer some of these questions. So this is indeed the same yeast that we use to make bread and we use to make beer, and it turns out to be an incredibly powerful tool in lab for a number of reasons. First of all, they share a surprising number of processes with people, little teeny tiny single cells. Many of these processes are essentially identical, including the SDH complex. Overall, it's a lot easier, faster, and way cheaper to work with them in the lab. They divide very quickly, and it's pretty easy to manipulate their little genomes. And we can pretty easily screen for mitochondrial or SDH function based on what we feed these cells, and I'll talk to you guys a little bit more about that in a minute. So just to kind of illustrate their similarities a little bit, here we have 2 protein structures. This one here is the human SDH complex, where we have SDH A, B, C, and D. And then on the other side, we have the yeast complex, where we have SDH 1, 2, 3, and 4, and they look very similar, and they do very similar things in the cells, which makes yeast a really great model to kind of look at this. So in the lab, there's a couple of different ways that we can measure to see if SDH activity is actually functional in these cells, and we can measure it pretty easily in a number of ways. The first one is with an enzyme assay that actually changes color if the enzyme is functional. So we add succinate, which succinate dehydrogenase works on. We have a PMS, which is just another chemical that helps facilitate the reaction. And we have our isolated yeast mitochondria. And if the mitochondria is functioning right, and if our SDH complex is functioning right, we get a color change. And we can actually watch this color change, where first everything's blue, and then as the reaction proceeds, we see loss of color in wild type. We don't see a loss of color where we deleted the gene, so it doesn't have SDH1 at all. We gave it back, we restored the reaction, and down there are a couple of different mutations, all of which, I believe all of which have been ones that we've seen in patients that are the yeast version of the human mutation. So you can see there's a number of them that are loss of function, where we no longer get that color change. One of them that would be classified as a benign mutation, where we do get the color change. And then the last one, again, no color change, so we have something that's likely pathogenic. So we have a fancy machine that measures this color change, hands us a bunch of numbers. I do a lot of math, and we turn the pretty color change reaction into charts where we're actually able to do statistics and measure the significance of these changes. Along with the enzyme assay, we're also able to just measure growth based on what we feed these cells. Yeast are really cool in the sense that they can make energy in different ways. One of them, if we use glucose as a source of food, yeast do fermentation, which is how we get things like beer. They don't need their mitochondria for that. They don't need the SDH complex at all. They can just ignore it. So we see that everyone is growing. We have our wild type, which is our brand normal, healthy SDH complex. We have deleted SDH1. And then we have a couple of different mutations, some of which we know are pathogenic in humans, and some of which we know are benign in humans. If we change up their food and give them something like glycerol, where they have to have SDH to work or to live on this media, we can see that our wild type is growing just fine, but if we delete the gene, we no longer see any growth. With the mutations we know are pathogenic, again, we no longer see any growth. So we can pretty easily, with both either a color assay or just with this growth assay, see whether or not a mutation is pathogenic or benign in yeast, and that seems to pretty consistently translate to the human mutations. Which ones are benign, which will allow for, again, early detection, better screening, and hopefully will result in better treatment in the future for SDH deficient GIST.
And finally, in terms of SDH deficient GIST, we're making progress. So of the original 4 drugs that were approved, imatinib, sunitinib, regorafenib, ripretinib, in those studies, in some cases, we were able to identify there were SDH deficient patients, but in general, there weren't very many, like 3. And in general, the response rates were less than single digits in terms of people who derive benefit. So we've gotten smarter and we've banded together doing studies that are only for patients with SDH deficient GIST, because we realize that it's different. Originally, when we did our first study of lincitinib, people, even the National Cancer Institute said, you people are crazy. There's not enough patients. You'll never be able to do a study. Why should we even fund you? Because it won't happen. But we did it, and we got 15 people. The drug did not work. But we proved that if we work together, we can actually figure something out in a normal timeline of drug development. So since then, when we're doing our SDH drug development, we're no longer taking every GIST that walks in the door. We're saying if you have SDH deficient GIST, we have a specific treatment, and that's what we're testing. And by doing that, we've now moved, from basically 0 for most drugs up to in the 20% for some other drugs. And at the bottom, Suzanne George out of Dana-Farber ran a phase 2 study of an inhibitor called regorafenib that had the highest response rate ever shown for SDH deficient GIST, 42%, very impressive. The unfortunate thing is that the company making the drug, and again, they weren't trying to make it for GIST, they were trying to make it for other cancers like bile duct cancer and cholangiocarcinoma, things like that. They decided they didn't want to develop the drug anymore because they didn't see a path forward. So they said, you can keep doing until you run out of drug, you can keep treating patients. We are planning to use a new drug called pemigatinib, which is FDA approved. We're planning to do a study for SDH deficient GIST. And once we can kind of work out all the funding stuff, we actually got positive on the funding. We just need to figure out logistics. We expect that to happen first quarter next year. So lots going on. More things are going on now than ever before, or even cumulatively, everything before is less than what we're doing now. We're doing our best. This is our mission. Thank you.
And with that, again, I would like to thank you all.